Actually, where we're at, they're trying to put us all on Outlook.
Oh,
They're trying to have us like really take advantage of everything on Outlook, and it's like really challenging because
mhm.
I mean, I'm sort of good with the calendar and all this, but we have a lot of media sites.
Well, I can go ahead and get started. Let me just share my screen. All right. So my project is on how leptin signaling is able to drive tumor growth in triple negative breast cancer. And I always like to start off by talking about why my project matters. So, as everybody knows, obesity is becoming more and more prevalent in the US and all over the world. It's a highly complex and progressive disease that's profoundly affected by the social determinants of health and structural inequity. The World Health Organization estimates there will be more than 1 billion people with obesity by 2030. And in the US, obesity is especially prevalent in the South, and Louisiana, where I'm at, has the second highest rate of obesity in the country, with 40.1% of adults being obese. Um, as you can imagine, this has critical implications for health, as obesity is associated with an increased risk of developing many diseases. Most people think about diabetes and cardiovascular disease, but it's also associated with an increased risk of developing several types of cancers, including breast. In the US, obesity related cancers account for 40% of cancer cases, and obesity is quickly overtaking smoking as the most common modifiable risk factor for cancer development. In breast cancer specifically, there's evidence that obesity can drive carcinogenesis, as an increased BMI is associated with more aggressive tumor types and a higher risk of recurrence. This trend is also seen in a more clinically aggressive subtype of breast cancer, triple negative breast cancer, or TNBC, with meta-analyses reporting between a 43 and 80% increased risk in developing triple negative with obesity in premenopausal women. Additionally, TNBC patients who are obese are more likely to have poor outcomes. They're more likely to have larger tumors and positive lymph node status at diagnosis, as well as higher mortality. So for these reasons, it's really critical for the complex mechanisms through which obesity is driving these poor outcomes in triple negative breast cancer to be understood. So one of the potential drivers of these poor outcomes for obese triple negative breast cancer patients is leptin. Leptin is an adipokine expressed proportionally to fat mass, and it plays a critical role in the endocrine functions of adipose tissue. So expressed proportionally means that if you are obese, you have hyperleptinemia, you tend to have more leptin present. So it's a peptide hormone, and it acts through leptin receptors in the hypothalamus to regulate energy balance through suppressing appetite and regulating lipid and glucose metabolism. But leptin receptors are present throughout the body, and there are 6 different isoforms of the leptin receptor, with the full-length isoform acting through its canonical pathway, the JAK-STAT pathway. There's also evidence of non-canonical signaling occurring through PI3K, AKT, ERK, and the MAPK pathways. Failure to produce functional leptin hormone results in hyperphagia and obesity, as well as immunodeficiency, as seen in the leptin-deficient OB mouse. You can see here a graph showing weight gain in OB male and female mice compared to mice with one functional copy of the leptin receptor. So leptin signaling is really relevant to breast cancer due to its presence in the tumor microenvironment. It's an adipokine, meaning it's produced by adipocytes and adipose stromal cells, which are present in the breast tumor microenvironment. In obesity, ASCs become altered and produce higher amounts of leptin, as well as other cytokines and adipokines. And both leptin and its receptor have been found to be associated with poor outcomes in triple negative breast cancer. So if you look at survival, high expression of both leptin and the leptin receptor is significantly associated with reduced survival in triple negative breast cancer patients. And together, all this data really has been the foundation of my project, which is studying how leptin from obese imprinted adipose stromal cells is able to affect triple negative breast cancer cells. In the Burrow-Collins-Burrow lab, we use patient-derived models. I know the slide seems like a little bit out of nowhere, but it's relevant because these are the samples that I was able to give y'all. So we take a piece of a patient's tumor, and we're able to propagate that in mice as a patient-derived xenograft. That's what our 4IC tumors are that I performed the experiment I'm going to talk about later is. I'm also able to maintain some of the cells from those tumor in cell culture and use make a patient-derived cell line. So we have both a tumor and a cell line from our 4IC. We have our 4IC tumor and our 4IC cell line. And then I can also culture those cells three-dimensionally to make a spheroid. And I'm starting to get into that, and I'll show a little bit of spheroid data at the end. This is just showing that we have that of the triple-negative breast cancer cell lines and some ER-positive cell lines that we use in lab, the leptin receptor is present. And then to study leptin signaling in vivo and in vitro, I'm using Aloaca, which is a small peptide pharmacological inhibitor of the leptin receptor, abbreviated AA. So this is the experiment that we collaborated on, that where I sent you the legs from. So the way I set up this experiment was in order to examine the effect of obesity and hyperleptinemia on tumor growth, I took skid beige mice. So these were immunocompromised mice, and I fed them either a high-fat diet, which had 60% kilocalories from fat, or a standard laboratory diet. I kept them on these different diets until obesity was achieved. I defined obesity as greater than 30% weight gain. Once this happened, I then took a piece of our TUBCX4IC tumor and implanted it in the fourth mammary fat pad of these mice. Then I allowed the animals to recover for 12 days, and then I began treatment with either Aloaca or with our vehicle, which was PBS. So at 12 days post-implantation, so this was when I started treatment, the tumors were significantly larger in the animals that were at a higher weight. So this is showing that the animals that were on the high-fat diet actually are heavier than their normal diet peers at this point. And this is showing that the tumors are also significantly larger as well. And this is when I began treatment with my leptin receptor inhibitor. So this is showing changes in tumor growth over time. I like to, when I present this, break this up just to make it a little bit more clear. So this is showing that in the high-fat diet animals, their tumor growth rate was significantly accelerated compared to normal diet animals. And I like to keep my high-fat diet and normal diet controls on the graph at all times so you can see where the treatment groups fall relative to that. When I treat with my high dose of Aloaca, that tumor growth rate is able to be pushed back down and is significantly different compared to the high-fat diet control. At my low dose of Aloaca, it's actually not sufficient to slow that tumor growth rate down. To me, that makes sense because in a high-fat diet context, you have hyperleptinemia. It makes sense to me that you would need more leptin receptor inhibitor in order to swamp out the effect of all that extra leptin. However, in my normal diet animals, that 1 mg per kg dose of Aloaca is sufficient to slow the tumor growth rate down. We also see that survival is affected by obesity, with mean survival being significantly longer in the normal diet animals compared to the high-fat diet animals. We see similar, though non-significant, trends with the treated groups. Interestingly, the mice are able to maintain their body weight for a little bit longer. One thing that happens with this type of experiment where you let the tumor grow to a defined endpoint, usually, I think with this experiment, we set that endpoint at an estimated 450 millimeters tumor volume. So that's a pretty large tumor, and these animals get very sick towards the end. They lose weight, they get cachexic. And you can see here in the treated groups that that body weight, they either continue to gain in the case of the obese animals, or they maintained their body weight for longer. This makes sense because leptin receptor inhibition stimulates appetite. We know that from the OB mice. But I also think there might be something happening with inflammation as well. Cachexia with cancer is more than just loss of appetite. It's a process where you lose appetite, but you also get this distinct pattern of muscle wasting and inflammation. So I wanted to look at the gene expression in these different treatment groups. So we sent off both the mammary fat pads, the contralateral mammary fat pad, as well as the patient-derived xenograft off for RNA sequencing, and then I ran pathway analysis using Enricher. I'm just going to talk about expression differences in the PDX tumors and mammary fat pads in the high-fat diet compared to the normal diet, and the high-fat diet compared to the 10 mg per kg aloaca. Apologies, this should say 10 mg per kg instead of nanomolar. So in the normal diet compared to the high-fat diet PDXs, we see a little over 2,000 differentially expressed transcripts with differential expression of genes related to inflammation, cell motility, and epithelial to mesenchymal transition. Pathway analysis suggests that these genes may be under regulation of the IL-1 signaling pathway as well as the VEGF signaling pathway. In the high-fat diet aloaca treated compared to the high-fat diet control, we see around 600 differentially expressed transcripts with differential expression of genes related to the extracellular matrix and EMT, as well as inflammation. So by comparing these data sets, we can determine leptin's contribution to the high-fat diet phenotype. Essentially, you have any genes that are in the aloaca group, but not in the shared group, are the changes due to leptin signaling independent of the high-fat diet. The genes that are in the normal diet group compared to high-fat diet, but are not in this middle here, those are changes due to the high-fat diet independent of leptin signaling. So we know that obesity causes many different effects and differential expression of many different cytokines and adipokines. So this is everything that's happening independent of leptin signaling. And then by comparing the overlap between these two data sets, we can see what's happening in the high-fat diet phenotype that is due to leptin. And we can look at both upregulated genes and downregulated genes. There are 124 identified genes that were similar here, with transcription factors suggestive of pathway, including NF-kappa B and EMT genes like TWIST1. I also want to point out PPAR gamma up here is another transcription factor that is suggestive of being a relevant transcription factor for control of these 124 identified genes. I also looked at the mammary fat pads of abbreviated MFP. This is not as much my area, so I would love any interjection here. But I see a lot of genes related, or at least Enricher. So Enricher is identifying these genes as being involved in citrate cycle and then different metabolic changes in cell metabolism. We also, and then it also identified PPAR alpha as a gene that is potentially, that these changes are potentially under the regulation of. In the high-fat diet aloaca versus high-fat diets, we see only 640 differentially expressed transcripts, with most of these changes being related to cytokine signaling and inflammation. But interestingly, I saw this gene, protein peroxidase pathway 2, being one that was potentially relevant in this, in these differentially expressed, in regulating these differentially expressed transcripts. This is just showing the RNA sequencing that was performed with and identifying genes that were differentially expressed under these conditions. I also alluded earlier to this muscle wasting phenotype. So when I showed the weight data, I was seeing that when you just look at the data, the aloaca treated animals maintained their weight for longer. But even when I was looking at animals that had the same weight at endpoint, so these are two mice that were sacked on the same day. One of these is a high-fat diet control mouse, and the other one is a high-fat diet aloaca-treated mouse. So these mice had the same body weight and the same tumor volume, but the appearance of the mouse is like very drastically different. You can see here the top of the hip bone, the limbs look a lot thinner, so does the tail. You can see the ridge of the spine. And one of the ways I talked to the vets about scoring the body condition of these mice. And basically the way to do it is you score them from like 1 to 5 and have the vets do that. But to me, this really illustrated that maybe there's something else going on beyond just the body weight and the tumor volume, that there perhaps is some kind of muscle wasting happening here. And this is why we got in touch with y'all and wanted to send you the legs to see if this was something that could be measured through metabolic changes in the muscles and in the bones. So now I'm switching to some of my in vitro work. So I wanted to look at how leptin receptor inhibition affects expression of EMT genes as well as tumor initiation capacity. This is just PCR data showing altered expression of EMT genes in 4IC cell line and also MDA-MB231s. I also see a reduction, though non-significant, in sphere forming capacity with aloaca treatment, indicating a reduced tumor initiation capacity with leptin receptor inhibition. Given the effects I saw on tumor volume, I also wanted to look to see what leptin was doing with cell proliferation. So I took three of our different triple negative cell lines and treated them with either leptin or aloaca, and then studied their growth over 5 days, DAPI stained, and then quantified that. This is showing that leptin signaling has no effect on proliferation in vitro, in 4ICs, in 231s, or in HS578Ts. So given that the effect of leptin signaling doesn't seem to be proliferative, I'm wondering how my tumors are able to grow, how I'm able to see this pretty strong effect on tumor growth in vivo. And what I think is happening is that it's an angiogenesis related effect. So I wanted to look at other genetic changes associated with the leptin receptor, and to do that I used GeneX Minor, which is a breast cancer gene expression miner of transcriptomic data. I did unbiased exhaustive correlation mapping, and then to condense and refine it, I did association of highly correlated genes with the removal of non-coding genes, which is what you see here. And genes related to angiogenesis and proliferation of endothelial cells were top hits. This makes sense given the association between epithelial to mesenchymal transition and angiogenesis. DXCL8 came up multiple times in my RNA-seq, and that's also a well-known inducer of angiogenesis. So given this, I'm in the process of looking back at my PDX tumors for protein expression of angiogenesis markers. I'm looking at CD31 or PCAM. It's a very well-established marker for endothelial cells. This is what it looks like in healthy vasculature. If you've taken a pathology course, this is what you would expect it to look like, where you see really strong labeling within the lumen of a vessel. However, in tumors that are highly disorganized, or tumors are highly disorganized, this is what it looks like, where you see more dispersed staining. And this is mouse breast cancer. So this is what my PDX tumors look like, and you can see here I have higher expression of CD31 in my high-fat diet tumor samples, but a reduction in that staining in my aloaca treated tumors with a similar trend in reduction in normal diet compared to normal diet treated as well. Next, I started to look into spheroids, and I wanted to show just some very quick initial data on spheroids. So I've been culturing our TUBCX4IC cell line three-dimensionally. We had a cool demo of a microscope, and I wanted to use that as an opportunity to see what the effect on cell death has been. I haven't been able to, just as I haven't been able to show any effects on proliferation in vitro with aloaca, I've also not seen any cell death. Spheroids, however, are a lot more similar to tumors. They have biologically relevant diffusion gradients, they have central hypoxia, and they're a lot more likely to be similar to what I would see in my tumors. So these are just showing my spheroids. Here's a control, and then here's an aloaca treated. I actually had a bunch of spheroids, so I also treated with paclitaxel as well as paclitaxel and aloaca simultaneously to see what the effects of these two drugs in combination would be. These are just two extra drugs that we're using in another project. I had an extra row, so I threw them on. But what I wanted to highlight here is that in my aloaca treated spheroids, I don't really see any differences in the amount of live cells. So live cells are labeled with calcium AM, but I do see an increase in ethidium homodimer positive cells. This is a dye that labels DNA, and really this is used as a live dead stain to quantify cells that are dead. The microscope I used had a cell-based analysis, so in order to quantify those. So I wanted to show that with aloaca treatment, I do see an increase in cell death. So that could also be an explanation beyond the effects on angiogenesis for why I'm seeing some changes in tumor cell growth. To conclude, this is my proposed model for my project where obese imprinted adipose stem cells and adipocytes are making more leptin, and then that's driving angiogenesis and EMT and metastasis in triple negative breast cancer. And I shout out to everybody in the Burrow Collins Burrow Lab, and please ask me any and all questions about my data.
Hey, Courtney, thanks for your presentation. I just a few quick questions. I might have missed it during your presentation. So the tumor you planted on the mice, it was TUBCX4IC, right?
Yes.
Okay. And can you please share the biological sex of the mice that you sexed?
Oh, of course. These were all female mice. I'm sorry, I should have specified that.
No, it's fine. It's fine. And the age of the mice?
So we ordered these mice about a month before I started the experiment. They were on the diet for about a month before I started the experiment, and then at endpoint, so they were probably 6 weeks old when they arrived, let's say 50 days when I began the experiment, and mean survival for so the oldest would have been 3 months. 6 weeks.
So when you sacrificed the mice,
Yeah.
did they die naturally, or you decided based on the tumor growth and size?
This was based on the tumor growth and size. We can't allow the tumors to grow beyond a certain point just because of IACUC. I set that limit at 450. microns, and I measured both the. So I talk about it in terms of tumor volume. When I was doing this experiment, I just did an X and Y measurement. I didn't do a Z measurement, so it's really more of an estimated tumor area. But
Yeah, I think you showed a graph. You shared a graph with us where we can see the tumor process. Okay, thank you.
yeah,
That was really interesting, and I was really astounded at just how the animals looked when they had the same weight and same tumor burden, but they looked dramatically different. So to me, since we're kind of moving into muscle, the whole muscle wasting inflammation obviously must maybe playing a role. Can you because I was trying to figure out what you were saying about PPAR gamma and PPAR alpha. That comes up. PPAR alpha is coming up more in the bone conferences. Like everyone studies PPAR gamma because of the glutazones and regulating diabetes, and that's why we were targeting it. But what specifically did you see with you said a PPAR gamma and a PPAR alpha effect specifically?
Yeah, so I need to spend more time with this because I'll be very honest, cell metabolism was never my strongest suit.
Oh, trust me, we are like, and because we're a melatonin lab, there's a mitochondrial melatonin receptor, so that's why we're starting to, because everything we're looking at is pointing to this PPAR gamma switch between the osteoblast and then the adipocyte, and now it's between the osteoblast or the adipocyte and the myocyte, and it's like the major controller of so many fates of a mesenchymal stem cell. So it doesn't surprise me that it's in here, and a lot of your pathways are pathways that we also see. Like, so we have this whole inflammation pathway. That's why we're trying to fold it into something, a paper. Like, so it's completely dovetailing nicely with what we were seeing, just with the few proteins that we were measuring. So my first question is PPAR gamma, PPAR alpha, and just do your best. Like we're all just discovering this as well. But the other thing, and I want Afsana and Young to chime in, but what you saw with the high dose AA, only in the high fat diet, and then the normal dose, or the lower dose, 1 milligram AA per kg, in the normal fat diet, we saw that also. And I'm trying to remember though what, I think it was bone, Young or Afsana, help me out with that one, but remember we saw significance for the low dose in one of the proteins in one tissue, and then we saw only an effect of the 10 milligram per kg. You guys try and remember your data. But anyway, Courtney, why don't you just go ahead and just first tell us about the PPAR gamma, PPAR alpha.
Yeah, so basically as far as I've gotten with that is looking at my RNA-seq and then running it through Enricher, and this is looking at the ARCHES F4 transcriptomic thing. Basically, you take your list of genes that you know are different in your data set. So this is, the PPAR gamma comes from the 124 genes I identified as being similar between my two data sets, where this is the normal diet compared to the high fat diet, and then also the high fat diet treated compared to the high fat diet. And then by looking at overlap, I can see what in the high fat diet phenotype is due to leptin. I put in those genes.
We do this all the time because you want to see what's common between both and then what's unique, and you want to go for these genes.
Totally.
But do you remember if PPAR gamma was up or down in the genes? Because sometimes that doesn't even matter. You have to do the proteins.
Oh, yeah. I don't remember, but I can find out and email you.
Go look that up. And then what I was going to suggest, if you have any more mammary fat pad, why don't you send it to us, and then we can look at the proteins we look at, because we look at, some of the proteins that you're showing and the signaling pathways, and we can just validate and just see how they relate to the muscle.
Yeah. Would you want frozen?
Yeah, if you have anything frozen, that would be great. Because we're just screening and running a whole bunch of these assays. We look at fat in a different model system as well, and we look at bone and muscle, but you might as well have us just look at the mammary fat pad to look at some of the things we're looking at. And we'd be happy to do that. But if you can follow up with us and let us know what is happening with the gene levels with PPAR, gamma, and alpha, because we were going to start, when I was at our last bone conference, I said, oh my gosh, they're all starting to look at PPAR alpha, and I'm trying to remember why that was. Afsana, do you remember what it was about PPAR alpha? You're on mute.
So PPAR alpha was mainly responsible for, I think, in the muscle for insulin sensitivity. So when they were looking into metabolic markers, PPAR alpha was coming up frequently.
Yeah, and so this is all relating, obviously, to the whole insulin sensitivity thing, because you can look at insulin in a couple of ways. You can become resistant, and that's what's leading to all the problems with the high glucose and throwing off adipokines. But also, insulin is an anabolic agent, so it can be useful, sometimes for building tissue. But then it can also, if too much of that and you cause the insulin receptor to desensitize, then that could be problematic. So that's something I said to my group. I said, we should probably start looking at this other isoform of PPAR. So, but I was so struck by just the difference in how your animals looked.
Yeah.
And immediately, and I just appreciate the whole thing when you're looking at cachexia, because my husband's a nurse, and so he sees how horrible that is for patients. And if you can keep them eating and, just keep some weight on them, that can really improve their quality of life. So but what I'm impressed with is just how much great overlap there is. And I don't know if Matt's on here, but we really have a lot of this is another system that we're looking at, but so many of these inflammatory markers pop up in everything we see. And even though it's the last thing I want to get into is inflammation, because I'm like, everything I do, I'm like, oh gosh, now I got to study these metabolic proteins. I'm a biochemist by training, but it's like, I keep having to do more and more, and I'm like, I got to learn about these. So, but I think what you're showing is really interesting, very novel, and it's really tying in the whole impact of obesity and leptin, and they're kind of showing different effects. And you can, and then you develop this novel therapy based, for cancer treatment. And I don't know if you follow Ozempic, that's the big craze right now. And what's the issue with Ozempic is even though it reduces weight, you lose lean body mass.
Yes.
That's a problem with that. Is I don't want to lose my muscle to lose weight. You want to lose your fat.
Yeah, that's really interesting.
Yeah, you lose lean body mass, and then you also, in a lot of, not a lot, but there's very severe adverse effects of it, because people can get literally complete deadening of their GI motility. So can you imagine that?
Well, that's what killed what's her name, right? Presley's.
What?
Oh my gosh, are you kidding?
No, they said that she had she was on Ozempic, and then she had a complete obstruction, and then I think her bowel perfed.
Wait, who?
Oh,
Priscilla Presley's daughter.
Yeah, Priscilla. Yes, that was Presley's daughter.
I did not know that. So they're downplaying that, and and I teach, about these diet.
Search it real quick to make sure I'm not thinking of somebody else.
But see, they those are big problems, and the whole thing with the lean body mass is, we study it from obviously a bone point of view, and we never really looked at muscle until we started, you're thinking, well, muscle pulls on bone, and you got to have strong muscles to cause a tension on the bone to keep your bones healthy, and you cannot, those have to be, studied together. And then you guys, we're bringing in the whole diabetes thing, because diabetics and their bone loss is different. Then you bring in all the fat contributing to bone loss, which is dramatic because of inflammation. And it gets down to very similar gene pathways that you're showing, it's like I've seen a lot of these in my work. And for the longest time, I used to study, breast cancer and the bone parallel, but now they're always like merging because they share pathways.
Mm-hmm.
That makes sense, right? Because our bodies are very integrative. So I guess if, I think from what I'm looking at, we should probably explore a little bit more of the muscle thing. We could do that, and also maybe look at some of the proteins, the metabolic proteins that we were looking at. We can just take some of your mammary tissue and look at it if you want.
Yeah, that would be fantastic. Would you want mammary fat pads from all of the treatment groups, or would you want just these main? So I guess this would be high fat diet, normal diet, and then high fat diet treated.
Yeah, I think. Well, let's have Matt weigh in on this. Sorry, my TV just went on. And then, and Young and Afsana, can you tell me do you remember what the normal diet animals where we had a significance with the AA?
So that one was the bone sample. The protein mark was the third one. So in the aloaca treated one, the third one level went down.
Yeah, so we're having SIRT1 and
diet.
Obviously it's big protein and muscle. But there's where we were showing an AA effect with SIRT1, which that's involved in a lot of these important pathways that you're looking at. It's not just a muscle protein, it's a protein involved in a lot of these, even inflammation pathways. So that's one of our targets that came up from our long non-coding RNA data. And then what else? Anything else come up with just that showed the 10 mg per kg?
In uncoupling protein 1, only 1 mg per kg reduced the uncoupling protein 1 level in the high fat diet group.
Versus normal?
Versus high fat diet control.
Okay, so high fat diet versus normal diet, only the 1 mg per kg, but you still had a low level in the 10 mg per kg. It just wasn't significant, right? Because you had a little variance.
Yeah.
So, Paula, I'm going to have to leave soon.
Okay.
I have a few more minutes. I have maybe 5-10 more minutes, but in terms of the samples, I think we can figure out what you all need to do samples, with the fat that Courtney's not going to need for anything else. I think my only consideration, Courtney, would be, are we going to have enough sample if you need to do any follow-up, with your samples? I mean, the other option would be, Paula, is if Courtney has a couple of lead hits from the RNA-seq, and if your group's already going to isolate the protein, to get maybe the Westerns. And then that sort of brings up, I think, Paula, what we need to try to do is define how we're going to look at papers from this. Because, I mean, you already have a clear, you've got two people that have done a lot of work on your end already. And do you imagine some of this going into their paper? Or, I mean, obviously we would have authors on, if any of this, obviously, but we need to figure out how we would kind of break this into a cachexia bone paper, and then the other part is the cancer, that kind of. So I think we need to figure that out too, because I think that packing it all into one paper would be sort of overwhelmed, because in some ways what Courtney's got is this anti-cancer effect of the drug on high-fat, low-fat diet, but you can't disentangle the tumor aspect of it from the cachexia high-fat diet, low-fat diet Westerns that you're running. So one option may be to try to take, like you suggested, the mammary fat pads and some of the RNA-seq with that and tie that into the cachexia bone muscle story, or break those two apart however you all want to. And then, that's sort of, we become part of that one. And then you all will be part of, Courtney's cancer one. Does that, I mean, I don't have the figures yet in my head. I'm trying to work on it.
No, and that's, because we were using these kind of as rationale as to why are we even studying? Because my students have all these whole different other projects.
Yes.
We were trying to lay down the foundation. So yeah, like we would definitely, we can fold everything in together for sure.
Yeah.
We can elaborate more on this.
Yeah.
Okay.
I also have some proteomics that I'm sending off in the new year with obese ASC spheroids and then 4IC spheroids treated with aloaca. So hopefully those would provide some proteomics data, in vitro rather than in vivo, but on leptin receptor inhibition in an obese context in another way as well. So we could see if the same hits are coming up in 3 different data sets, your data set, the in vivo, and then the in vitro spheres.
No, that would be great. And like I said, we can, whatever, yeah, you keep your tissue. Like, we don't want to take, they're not that big. So we've worked with mammary glands, or fat pads before. So,
well, what I would say is maybe what Courtney can do is, she and I can maybe next week or then over the holidays kind of come up with an outline of what she's thinking about including in her paper, and seeing ways in which the data, from your end, could benefit, or there are experiments that may not be bone and muscle related, but a Western blot, in a different tissue, a tumor tissue, or the mammary fat pad that would add to the cancer paper. And then we can see what data, like for example, the RNA-seq or other things that might be useful to merge into yours. And that way we she has an outline of what we think we need for, the primary paper that she started. And then that way we see what else we can craft out of it. Because obviously your students have their projects and this can be part of it, but I don't want to just, I want them to get their credit and get their papers.
oh, of course.
Oh,
Yeah.
absolutely.
Yeah.
All right. But before you go also, can we somehow talk about the tumor studies in C4, C5, melatonin, tamoxifen drug conjugates, or?
Yeah, we can. Sophie?
Can I ask a question real fast before we move off Courtney's stuff? Courtney, I couldn't remember. Did you ever keep serum from the mice?
No.
Okay. I was just thinking because of all the inflammation that kept coming up. I know that with bone and adipogenesis or those transitions, the inflammation levels matter. So I just didn't know if that was something that we could look at, because I know you saw IL-8 and stuff like that, but we don't have a serum or anything like that, so we can't detect the levels that was between the different mice.
Yeah, I wish I would. That's such a good idea. I wish I would have thought about that at the time.
We always do cardiac puncture, take that, make serum, because we always do ELISAs when we can. So, because, inflammation, then you can quantify everything.
Where I am wanting to do conditioned media ELISAs from some of our obese breast-derived ASCs. It's not the same as
It was just a thought.
Would it make sense to do maybe some diffed ASCs and then conditioned media from those before and after Aloaca? Would that? I'm just thinking for cachexia paper.
It'll probably be too artificial, and I'm not really. I don't know enough about cachexia, but I know with the bone and stuff like that, if you had high IL-8, you could be pro-bone, whereas medium levels of that could be anti-bone or something like that. So I was just thinking that with some of the stuff you guys were looking at, if there was a tissue forming, the serum levels might help dictate, even if it's high or low, why the phenotype was what it was. And I don't know what leptin levels do, if leptin does similar, but I know the inflammatory guys, mid, high, or low, will push one way or the other for tissue function. Okay, sorry.
No.
For the mouse experiment, I have not done any pilot studies in the D538Gs, so in January, that's what me and one of the PhD students are going to start. So I'll do that. The parental cell lines, he is also going to work on doing a scaffold model for that, so that we can just see if they'll take off, how they'll take off best, plus or minus estrogen as well, since they are MCF7 cells. And then from there, once we establish what will work best, then I'll move forward with doing the drugs for the C4 and the C5 compounds.
Oh, that's good. Can you possibly just give me a little summary? Because I have to kind of update, I have to keep things straight in my head. So, because I have other people that are kind of asking about these studies and just what's the potential for, generating preliminary data for grants.
Mm-hmm. Yes.
We're moving, we have a lot of nice data on these, and that this is the next best thing. So would you be?
Yeah, I can absolutely write out all my experimental plans and everything and deadlines and just send that all off to you.
Or just tell us what you like in general. You don't have to make it a big deal.
Absolutely.
Just so I have to keep notes now.
Right.
I got so many different things going on, but I'm very excited about all of this. I think it's fun. I love collaborating and merging ideas. That's how things expand, because you need a lot of different eyes on the same thing, and it's like, oh, I didn't notice that.
Right. And then hopefully if that takes off, I've also written down the notes of all the areas of like the fat pads that I'll take for you guys that I can send to you if you want.
Yeah, that would be wonderful. We love fat, right, guys?
Yeah. Happy to do it.
We're a little slap happy. We were in the lab last night until 10:30, taking all of our clinical trial urines and freezing them down and sorting them. So I think we did like 380 samples, including the how we doubled it. So we're
oh my gosh.
Gotta go. Right. Thanks everybody. Thanks Sophie for putting it together, and we'll proceed. I guess we need to set up another meeting, early January?
Mm-hmm.
Yep, sure. And Courtney, that was awesome. So great work. Yeah, so keep in touch if you guys, if you guys can send me some things, and then we can just follow up.
Okay.
Yeah.
Yeah, that sounds
Absolutely.
good.
Bye, everybody.
Sounds good. Bye.
Bye.
Aww.
Aww.